Excited to introduce Dr. Singh for today's seminar. So Dr. Singh received his PhD from the National University in Singapore in biophysics and quantitative imaging, where he developed fluorescence cross-correlation spectroscopy and selective plane imaging microscope. So this is this idea of understanding the diffusion of proteins, subcellular localization of diffusion of proteins using the kind of fluctuation of fluorophores attached to those proteins, and combining that with kind of these newer microscopes that allow long-term imaging and kind of deep imaging using these kind of more selective imaging microscopes. So that's kind of this really nice application. And then after receiving his PhD, he went on to work as a research fellow in Dr. Timothy Sanders' laboratory, also at Singapore, where he built tools to study how neurons and glial cells robustly shape flying nerve cords during embryogenesis. And then he traveled to Princeton in the United States, where he's a postdoctoral fellow in the Louis Siegler Institute of Integrative Genomics. And his current research focuses on building quantitative imaging techniques and perturbation tools to dissect temporal details of cell fate decisions during embryogenesis. So I'm really excited about his talk today. He kind of works right at kind of this really interdisciplinary nature where kind of the field of developmental biology is pushing towards kind of right at kind of biophysics and developmental biology and cell biology, and so really pushing that edge. So I think we're going to hear a really excellent talk today. I just wanted to mention that if there are questions online, you should be able to ask in the Q&A, and I will ask them to the speaker when it's ready. Or if there's issues, please put it in the Q&A, and I will monitor it. Like you can't hear anything, let me know. And then after the seminar today, there will be kind of an informal meet and greet in Shoemaker 219. Come talk about what you've heard and meet Dr. Singh. So great, thanks a lot.
Thank you, Josh. Thank you for having the introduction and arranging everything. It's really good. Okay. So hi, my name is Alan Singh, and as Josh mentioned, I'm on the kind of interface of the biophysical tool development, and especially on the fluorescence-based methods, where we could actually look at the proteins, what they're doing, and how they regulate biochemical cells and the mood, and especially all these dynamics, how they're occurring inside the cells and driving the development of a living organism. And so today's talk is going to be built on the in Drosophila embryo, so early 3 hours of development. And so the protein called decoy, which is a morphogen. I'll go more into detail what these morphogens are and how they work. So I'm sure here everybody's kind of familiar with what are transcription factors. And so transcription factors are those proteins, they localize in the nucleus and bind on the certain sequences of the DNA for target genes and then turn on transcription. They first make mRNA and then they will be translated as a protein. And so here, this decoy is a morphogen, also a transcription factor, kind of, and it was actually discovered by Eric Michaud in late 80s somewhere, where they found out how these developing embryos get a polarity, how they start this very early stage where they have some kind of a polarity, where they can be anterior, they can be posterior. And so that's something important for establishing any given organism. You have to start with the head and tail for that given organism. And so now what we started looking into actually how this transcription factor that we can call as an input, that I'll be referring more and more. So that's input goes into certain regulatory connections called here gap genes. So these are the next level of information that is taken from the bicoid, and they express these certain genes called gap genes. So if I go into the detail, so this is the kind of a whole roughly 7 hours of development, and you can start actually seeing how the protein starts with a very coarse kind of like on the anterior it has a higher concentration, and then it kind of decays along the length. But in the next phase, it's a band, and these bands are details or expression patterns for proteins or transcription factors are getting refined and defined. And so how this is happening, and so one of the idea is actually in the development is these maternal genes, for example, here bicoid, works as a transcription factor. They will express the next set of genes called gap genes. And now, depending upon what gap genes are there in those body parts, they will further go into more refinement of a gene called PIM or many other genes. And so if we look at actually how these networks are connected to each other, so these arrows mean whether it works as an activator, and then aligned with the bar means actually it works as a repressor. So now you can see actually these regulatory regions, how complicated they are, and they actually change over time. So now you can see the maternal input bicoid, caudal, and nanos feeds into the next level of information called giant, tailless, hunchback, frugal, crowns. And we don't need to remember all these names, but just to give you an idea how these complicated networks are set to give rise to specific body pattern. So these are very complicated patterns, and they happen actually, you can see within 7 hours, you have many, many transitions. And so what we'll be focusing on these early 3 hours where maternal input genes are giving details about what genes or bands of expression should be on or off. And so one of the key idea in the developmental biology has been the French flag model, where what it says actually we have some kind of sort of a small diffusible molecules, and those are graded along the length of the embryo. So as you can see, this is graded along the length of the embryo, and depending upon the concentration of these small transcription factors or diffusible molecules can give rise to blue, white, or red type of cell. And this was actually before even bicoid or any given morphogen was discovered. So this was in somewhere late 1970s by Lewis Wolper. And so now you can see actually there are quite a bit of similarity if you compare the threshold from these morphogens to bicoid. Of course, this is a linear gradient, but that is a kind of exponential gradient where bicoid input transcription factor is graded along the length of the embryo. And what it's doing is giving rise to these, depending upon the concentration, giving an expression for a hunchback. So the region I'm interested in is indicated by that black bar, which is the anterior part of the embryo, not on the posterior. And the next gene is frugal. So now you can see it indicates a lower threshold value for bicoid to give rise to this expression. And the last one is the nerves. And so now you can see actually there are quite a bit of similarity. And when people saw actually bicoid and these gap gene patterns, they thought, okay, everything makes sense. Now we could understand what is happening. But we'll show some of the details. They are not quite make sense with respect to morphogen and these ideas. Okay, so if I go a little bit deeper inside the cell, what is happening? So now I gave you a very coarse information. Somehow these cells sense this transcription factor bicoid and give rise to expression for these bands of expression. So what is happening here, what people have in developmental biology have now created many of these details, what transcription factors are and how their expression is regulated. And so one of the detail about this transcription factor here shows is about this transcription of particular gene called hunchback. And so you can see that the green cell indicates where you can still have a lot of this decoy transcription factor. And of course, that recognizes specific sequences on the DNA called enhancers and turns on this gene called hunchback. And depending upon the concentration, as I told you, depending upon the threshold, where you have very few molecules available, this gene won't be on. And so with that, you can say, okay, now we have some idea, depending upon the concentration, we could predict how the pattern should look like and what should happen to this particular gene. But this becomes slowly more complicated because there is not only one given pattern of maternal input. You can see here there are multiple inputs from anterior to posterior. We don't need to remember all of these, but it gives rise to actually these complicated patterns of all the gap genes called giant, hunchback, frugal nerves. That's all. Or these are kind of segmented body patterns. And so if you have a mutant of any of those, that segment will be missing in those embryos. Of course, these embryos won't be live. They will. You can still understand what is happening at the body segment level, and that is very easy to do in Drosophila because you can make the cuticles and also look at the expression pattern. Okay. So now, what you can see actually the gene called frugal, which is the blue bar on the right side, and the nerves. Those are at a very low level of decoy over, you can see here, they come around roughly 50% where decoy concentration is very low. And now you can see these cells are not only sensing decoy, they also have other inputs called nanos or caudal or torsoline. So how these cells in a confusing environment determine whether they should be on or off and how these regulations are happening. And so what we are interested in to actually this time detail, the kinetics of these inputs. And it becomes actually very complicated to actually go understand what is happening where you have multiple inputs at the same time. And so you can see actually now you don't have only a given one transcription factor, that there are multiple inputs that can be also giving rise to this expression. And so quantitative biology is actually now trying to understand, many theoretical biologists or physicists are interested how this input, because this you can imagine as a cell as a small little computer or sensor that takes some information and gives out the output as an mRNA or protein. And so now how these cells, tiny little 10 micron, 5 micron system is able to do this computation. Also in a very noisy background where concentration can be very low. And so that's where, and so there are many challenges actually. I showed you a kind of a static picture, but we know development is not a static snapshot. It varies over time. Your input and output changes many levels. As you can see, all these gap genes are variable. So this is roughly runs for 60 minutes. So these are created by staining hundreds or thousands of embryos and aligning them in time. So these are dead embryos, but you take one embryo, you take one snapshot, and then you go on thousands of embryos. And that's how our lab actually created these movies from dead embryos. So that's how you can actually understand how these different levels of gap genes are coming up. And so there are two bigger problems with this. All these gene expression patterns shift rapidly on sometime on a minute time scale. And also they can regulate because these are transcription factor themselves. So they can also interact with each other in a way to give rise to the expression. And the second thing is, the last part is actually all this, in getting the details about how these genetic perturbations can be really slow. So if you want to understand how a given input is giving me the output for certain gene, and you have all these cross-regulation and also slow perturbation. So how you make this causality, any given change of your input is giving you the output. So this is very tricky problem. And the way we thought of solving this problem, so this gives you one of the gene I'm showing you here as a giant, which has, you can see, two bands on the right. It has the anterior band and then the posterior band. And now it's in the multiple these maternal inputs. And so one of the idea from the, so now since we know what are those maternal inputs, so we just asked the simple kind of a physicist approach, whether we could remove all these maternal inputs and this embryo becomes like a kind of reaction vessel, where we could analyze in a way where there are no boundaries, there are no regulation, that we could understand whether the change in given input will give me this output for the transcription. And so here, what you see actually, there are some of the details. These tiny dots, these are actually mRNA sensors. And so now in Drosophila, we could image these mRNA in a real time, which is really interesting. You can have an idea when that gene is turned on and progressing in the development for coming 1 hour or 20 minutes, whatever window they are on. So in doing that, actually, what we did, we removed all the maternal components like caudal, nanos, torsophyte, and big-white itself, but now put as a big-white is flat input. So now this embryo, which is a syncytium, all the nuclei are shared in a common cytoplasm at this given window, when we are looking at it. And what you see actually, what remarkable thing happens, there are no patterns left. All these giant expression for mRNA, you can see actually they are uniform throughout the embryo. So this becomes like a tool where these embryos are not going to survive, but they become a tool where you can investigate what happens when you change your only given input concentration and how the changes occur in the expression for your given gene. And what we are looking at actually multiple genes and how they are regulated. But in this case, actually, we cannot do any further because once you created one expression, you cannot change it. So they are fixed in time as well. So the next trick we used from optogenetics, where we could manipulate things with the light. And I'm just going to show you one of the movie. So now we created a uniform set of reaction vessels, not only one, but hundreds of those. They are doing the same thing. And now with the light, we could manipulate the concentration. You can see just flash of light. And so this runs on a minute time scale. You can see within a minute, it just comes back. If I want to give you an idea of how the difference is, just putting by blue light, this concentration of the new protein is decreased in the cell nucleus, and they are shared in a common cytoplasm. And so the way this tool works, this has this LOVE2 domain. It's a tiny peptide unit, which upon putting them in blue light, it changes the spatial conformation. And what happens is you have these certain sequences of export or import that will determine now whether that protein should be in this nucleus or cytoplasm. And that you can use, so now you can understand this is some blue light or white light here. By turning on or off with the, just simple switch, I can change the concentration. This is really something a developmental biology has not done yet. Or there are not tools available now, but by combining these tools together, some of these engineering tools, we could understand on a minute time scale what is happening, how these cells will respond not on the transcription factor level, but also at the output level. And so if there are any questions, you could ask.
What is the readout?
What's the readout here? Yeah, so what I have actually, I have tagged this picoid with the EGFP. And so now what you're looking at actually nuclear localized picoid. So these are individual nuclei, and they are in a common cytoplasm. So there are no cell membrane. This is a big ball of cell, you can say, and they are multinucleated. And as I turn on the blue light in this given region, this protein goes outside the cell. So that's what it shows here. And so with that, actually, you could create a kind of concentration difference between the nuclei here and the nuclei here. And if this, yes. Yes, so now with that, actually, you could do that. You can change the RNA sensor. So you mean to tag your RNA sensor?
So the previous slide showed
yes.
the apparatus, the RNA system.
Yes.
So over here, the synthesis.
You could use that, but idea is now in this way. Once you have control over your transcription factor, you could regulate your transcription. And so, depending upon the now concentration you have in these nuclei, so you have low concentration of transcription factor here, and you have high concentration here. So these are within a minute time scale you can do these modifications. So these are, so that's one detail. These are maternally loaded. So once mother lays all these eggs, they load all these mRNA, and these will be translated during these early stages before this stage. These proteins are already there as a transcription factor. Any other question? Stop me if I'm going too fast or not clear about the things. Okay, so now at least this gives you an idea. With these tools, I could do the manipulation on a minute time scale. And now look at how transcription will behave to these changes. Okay? So doing that, one thing I needed to build this microscope where we could do all these fancy stuff. So this is just normal human microscope. This is a custom built microscope. And what it does actually, it does like any given microscope. You take images, snapshots, but it also has this kind of a projector unit, something like we have here to project onto this screen. So it's very similar, but very tiny. And so I could create small features onto here, like I'm projecting my slide onto this projector, but now on a tiny scale of a roughly 0.5 millimeter size of an embryo. So on that size, we could create any given pattern. And so one of the advantage of using two-photon microscope is actually to illuminate only a region that you are sighting, not something in one photon, you excite the whole sample throughout. So when you scan many, many times, you bleach the whole sample much faster. So this is one of the key advantage of using two-photon. And on top, we added these tiny micrometer devices. These are basically hundreds of thousands of micrometer. And depending upon the image you want to project on, you could create any given pattern. And so that's what I was creating onto that previous embryo, and here you could create any phase or anything. And the second thing is you can temporarily modulate this concentration of a transcription factor or any given tag protein. So this is something when there was no blue light on, that's indicated by black. So you have more and more concentration in the cell nucleus. As you turn on the blue light, DMD is on, then you see the drop in the concentration. As you switch it off, it goes back again. And this happens on a minute time scale. So the export and import can be really fast. Okay. So with that tool, now we could reduce the photo damage, also have these perturbations on a minute time scale, and spatial precision. That is something, if I create these patterns on a one stripe of cell, it will kind of spread onto two neighboring cells. So that is something the scale of roughly 12 micron. And so these nuclei are roughly 5 micron in length. So this tool gives me very good hand on spatially on a certain part of the embryo, which is now homogeneous anyway. I could create perturbation on a given part of the embryo and see other part, how it's behaving over time with respect to your perturbation. So now you can do all these perturbations and unperturbed part of the embryo as a tool where you could read out these mRNA production, how they change. Okay? So what I showed you, okay. Sorry about this. Okay, somewhere. Comfortable. Let's try again. Let me skip the slide. No? Do you have any idea what's happening? As a PDF? Maybe I can close this. Go to, sorry about this. Downloads. Do you know where that was downloaded? So, nope. Sorry. Sure, let's save it. I think it's this. How do I go? I will download it. Okay, let me try it this way.
We're allowed to see some. Check that order with the double design in Michael.
It should allow me to play here. That. Okay. I mean, it's fine. It's just I can find this. Okay. Is it connected? Yes. Which one is? Yeah. Okay. Okay. So here, what I showed you, like how these complicated input of maternal inputs, we are looking into these specific gap genes called hunchback or Pupil 4 gap genes. And what we did in the next step is we removed all these maternal inputs in such a way that we left with a uniform set of information in these living embryos. These embryos are still live and they're doing all the kind of biochemistry for us, and but they won't be surviving as a living organism because they can't now reach to next level. Yes. So that's a good thing about Drosophila. There are all these mutants that you have to actually combine in such a way you have a lead out for your given input transcription factor, also these maternal mutants. And so if you want to know more, I can tell later how we do that. So basically, it's we have to prepare roughly 2 months of work to get to this point. Once we have that, then it's easier. And so now they became kind of a uniform set of details where concentration is uniformly expressed in these embryos. And also the output, now you can see there are no boundaries anymore. They are all uniform. And depending upon the concentration of your ubiquitoid, you will see specific combination of gap genes. But next step, what we did now, we implemented these optogenetic tools where we could manipulate them on a minute time scale and see how that change of concentration will lead to the expression change on a minute time scale, where we could also see mRNA sensors as a readout. And so this is on the minute time scale we could do by imaging and perturbing in the same given embryo. So that is something we try to establish along. And with this, why we want to do this 1 minute perturbation? Because we want to kind of remove all these other inputs. Those could be playing role for the expression for these target genes. And so the question we are looking at actually how these target genes respond to change of ubiquitoid concentration, and whether we could understand the kinetics and the strength of these mRNA production. And so that's a movie I'm going to play here. I hope it plays. So what you're seeing actually, these green little dots are mRNA sensors. These are individual nuclei. And the region we illuminated with the blue light, the concentration of your transcription factor is low. And the region we didn't put blue light in that given embryo gives me idea how much this transcription factor is regulating this gene particularly. So that's one given example. And so now we have idea about our output gene, which is tagged with those green little foci. Those are the mRNA for these target genes. And then we have also idea about our time about when exactly we did those perturbations. So just to give you idea aligned with the development of Drosophila, that is something happens over 3 hours of window where multiple rounds of these divisions will occur. And so basically it starts with the fertilized egg, it divides for another multiple rounds of 13 rounds of division, reaches to this point, and then it forms specific kind of these folding where all these surface cells will invaginate or called gastrulation and form different parts of the body. And so what we're looking at actually is roughly last 1 hour to 90-minute window where we do all these perturbations, and that's the real time window we are interested in. And so we can do these perturbations in those time scale around these. So these are indicated for different nuclear cycle divisions as number, and this is the time window of any given perturbation. For example, it can start somewhere cycle 11 to all the way to 14, or depends on those perturbations. And so, and all the analysis we are doing in this given window of cycle 14. And what you're seeing here, actually, these green, these dots over here, green on the right side, these are individual, now all these mRNA molecules for that given gene. And with the light, we could actually wipe out those dots depending upon the given transcription factor, how much we reduce with the given light. But this does not give us the kinetics. And so the way we did those kinetics is as follows. This will be something like you are looking at how many spots you have in a given ROI. So that changes over time of roughly say 40 minutes or 50 minutes. So this gray bar or the plot indicates unperturbed state. But there is no change with the light. These were kept in the dark. So what you're seeing actually, how the number of these mRNA spots increase over time. And now you can see this is something very similar, like you add a drug to your cell, and after a certain time, you observe what are the phenotypes happening. So that's the same thing we are doing here. We are counting number of I'm counting actually number of spots over time. So now you can see this was kept in dark, and around 11 to 12 minutes, I turned the blue light on. And you see there are fewer and fewer spots detected. And this change happened immediately. So now you can see how fast these number of spots are kind of different from unperturbed state. And also on the other side, here it starts again. So in this case, this was kept in continuously blue light. This will be my residual level. I cannot get rid of that many spots left. And as I switch from blue light to dark, what you see actually with the light, it was kept at low expression. As I'm turning off my blue light, what you're seeing actually the rise in the signal that goes along with the transcription factor increases in the nucleus. So that gives me idea about the kinetics of my experiments. And just to kind of summarize, I'm not going to go all the plots all together, just to give you idea what is happening with the time, response time we call it, how fast these genes will respond to change of transcription factor. You can see they're roughly within 5 minutes or 20 minutes. And there are two distinct states, you can say. One responds positively. That means when you bring back transcription factor in the cell nucleus, the transcription goes up. But in the other case, it becomes less and less. That is the negative mean. And here it shows when I'm removing with the blue light, so you see fewer spots. So lower concentration of picoid has a lower spot of this particular gene called hunchback. But in unperturbed state, you see all the spots come up very sharply at around 15-20 minutes, and then they go down. So this is the unperturbed state. And so in the next slide figure, you see the color or the blue one is flipped. As I remove this transcription factor out of the nucleus, the signal becomes very strong. So this picoid works as a repressor. That's what this negative means. And what I told you earlier, everybody thought picoid works as activator for everything. But here what we see in a very simple these perturbation experiments, this works as an activator as well as repressor depending upon the gene. So let's quickly go for these two. What this means, this is well established, well studied. We knew this, it should behave like this because it has these two known enhancers and it has lots of these picoid enriched regions. So picoid comes and binds to those regions. And so we knew as we remove picoid transcription factor, the transcription should go low. And that's what we exactly see. And that is also within 2 to 3 minutes, these genes will start responding to change of transcription factor. The other gene we looked at, called GIANT, also has picoid binding sites, indicated here, but it also has many other enhancers, those are non-picoid binding sites. And so that's why actually our response, okay. So, okay, that's good. Thank you. So here, what you see actually response between this blue plots and over here, this is much cleaner than this two. And the reason being actually, picoid is not only one given activator, and also GIANT is on the X chromosome. So you have male and female both contributions, and they work as a kind of a dose-dependent manner. So the response is not sharp as hunchback, but they all roughly are within 5 minute window, which is good. So this works as activator. And so now we kind of got further into the next gene, which is Prupal. And by doing the same set of experiments, either by shifting them into blue to dark or dark to blue condition. And so here in this case, you can see this is after roughly 22 minutes, you see when we switch from light to dark, the expression goes down. And so that's what indicated here as a 22 minute roughly. And the response is actually in light, you make more mRNA. That means at low concentration, you will make more mRNA of a transcription factor. And so we want to kind of get idea why this is this long delay. And so 22 minutes is long enough to make new product. And so that's what we started kind of got into. And so here what you're seeing actually 3 genes doing this in the same given perturbation. So here in the center part of the embryo, I put a blue light and keep them in blue light. And the region I put them in blue light, you remove transcription factor picoid. So there is low concentration here that gives low expression for GIANT. But at the same region, actually, you see Prupal is very strong. So, but for the other gene, which is hunchback, is hardly any change. So with this simple kind of a spatial perturbation, we could see how, so what is happening is you can see in dark, when there is no blue light, picoid comes, binds onto the regulatory region of GIANT, makes mRNA becomes protein. Now that works as strong repressor for this gene Prupal, and that's why you don't have Prupal in these regions where you have high GIANT. So this is particularly interesting. Now with this tool, you could find out actually how these nodes are connected in time and also that's relative strength. But in blue light, you remove most of the decoy. That's why you don't have giant, and the Drupal comes up. What is activating Drupal? We still don't know, because it was thought decoy should work as activator. So that kind of explains why we have this long delay of 22 minutes, because any given perturbation needs to go via giant for Drupal things. Okay, so I'm gonna go a little fast. This was particularly interesting. This is the most posterior part of the embryo for gene called nerves, and response is again fast but negative. And so you can see here, this is the unperturbed state where the embryos were kept in dark, and now as we put them in blue light, you see within 2 to 3 minutes, the signal goes really high. So the 3-minute window is not enough to make something new to have this expression over time. And so now this indicates this is directly Bitcoin is affecting the expression for this particular gene in a repressive manner. And this is a totally new detail that was not existed in the literature. And we still hope reviewers will buy these arguments. So to give reviewer a bit idea what is happening, so any given gene can be a combination of multiple enhancers. So we got further into those enhancers where this particular gene called CNURBS has 2 known enhancers called distal and proximal, and distal has more Bitcoin binding site than proximal. So Bitcoin can bind more onto these, and it's very easy to delete one of these or alter their function in a way you do not get the signal the same for your readout. So as you can see, as we remove the proximal enhancer, kept only distal, the behavior is very similar to this whole intact when you have both of these enhancers, but not in the other case where there is some temporal shift, but amplitude roughly remains the same. So now we know which particular enhancer is driving this change. Amplitude is different because as you chop off more and more region of your enhancer, you have less expression. But the time, you can see upon the time you start eliminating those embryos, within a couple of minutes you start seeing the rise in the signal. So now we know what is happening to this particular gene, and we could find out these temporal dynamics. So that was all good. So what about our morphogen that I started with? The different concentration of decoy should give rise to these genes, and but some of these new data actually indicate that's not the case. There are much more, and this wouldn't be possible by doing slow genetics experiments. And so now you can see actually, okay, so something's missing. So basically, Bitcoin works as the activator for these only 2 genes. It's delayed repression for this one, and it's fast repression for this one. But that goes totally against the idea of morphogen, where you have different threshold and everything works as activator for that given morphogen. So this is interesting, and we are hopeful viewers will like some of these experiments. So another 2 or 3 minutes I'll take to give a little bit more idea about how we could use this system of uniform set of embryos and change the concentration upon blue light. And so these light sensitive protein will change the concentration depending upon the intensity of the blue light. So that's kind of recapitulating some of these theoretical models into living system. And so this becomes very interesting and powerful, and to kind of investigate how these synthetic morphogen that you can manipulate over time and space to investigate these decision making. To do that, so this is the movie, this is the embryo I put on the flyer. So as you can see, when I put the cricoid tagged with Lexi in a kind of pattern of blue light, where it sees less light in the anterior part, which is here, and higher light on the posterior part, has more Drupal mRNA than the anterior, which is very interesting. This is totally not driven by the embryo. This is totally external input that you have given to embryo. And what is happening, as I showed you earlier, in that given perturbation of blue light, the gene hunchback remains throughout on. Giant becomes half the region you don't see. These spots will have higher giant, and the region sees more blue light has Drupal. And so with these, actually, we could investigate how these artificial gene boundaries that we could create just using blue light or some kind of a light perturbation. And so here we are looking into actually how this mutual repression between giant and Drupal works to define the boundaries. And this is crucial because that's how we get our digits or fingers well-defined boundaries or body segments. And this gives a very kind of a engineering way of handling these questions in a very robust and temporal manner. Just the very last slide. In the future, what I want to kind of go further about this hijacking the regulatory network. What I mean is, I showed you there could be multiple sequence of events that need to occur to happen before any cellular decision making. And now with the optogenetic perturbation, actually, we could cut out those regulatory region in a way where we could individually control specific pathways. And this was actually done at Princeton here. By using blue light, you could turn on a specific pathway called ERK, where just to give you a phenotype how it will look like, if you don't eliminate those embryos, they are lethal. And if you just eliminate on the poles of these embryos, these flies will hatch properly and will be viable adult by total external control. And so now people have looked at actually how this pathway is regulated and giving rise to these segments and the expression patterns and the kinase. And what I want to kind of further go into it on the JAK-STAT pathway, what it does actually, you have some kind of a external stimuli. Here in this case, in Drosophila, it could be a smoke event for larvae. And that actually triggers the immune reaction into those larvae or pupa. And one of the good kind of plus side about these pathways, these are highly conserved across the many organisms, and they are important in regeneration. Also, they are important in the normal development as well as the disease or the cancer. So this will allow me to actually investigate how these pathways can be activated on a something that you demand at a particular time in particular space of the embryo. And so this is something I could compare how these kinetics and the decision-making process happens over minutes or hour time window. So with that, actually, I'd just like to kind of summarize what I showed you. If it was not clear, we wanted to make a system where I could use an external stimuli and control a biochemical pathway. In a way, I could understand how these interactions are happening. And with that, we wanted to find out the kinetics. And so with the kinetic experiments, we showed actually how these are tightly regulated to control the expression for certain factors. And these perturbations we could do on the minute time scale. And now with the actually experiments with these target genes called gap genes, we found out actually the response can be from minute to half an hour window. So that allows now to rethink how these morphogen models and all these existing details were presented in earlier papers. And now, with the final thought, I would say now uncovering these kind of fundamental details about the cells, how they work, that could be interesting to know, where you have external control over system, and that you could follow over many time scale becomes very powerful to investigate these biochemical pathways. So with that, I'll thank all the university, my Eric Picoy, of course my lab member, my collaborators, and thanks for your time and attention. So if you have questions or something that you want to follow up. There were a lot of details I had to miss because it was a very long project actually.
Also, I have a question. Let me see if I can see if there's any online questions. But so you mentioned identifying Bitcoin as a repressor for genetics.
Yes.
So you identified maybe some cancer sites.
Yes. So now what we are looking into actually going. So this specific enhancer is still 2.3 kb long. And so now we are going to actually minimal part of it. So we made those lines, we're still trying to image those because still to get those embryos uniform takes a while. And so, but some of the experiments already indicate Bitcoin is not even required to turn on this gene.
Yeah,
you can have other gene called,
yeah,
there is something else that can turn on. And so Bitcoin works as a kind of anterior boundary repressor. And so that's what we're seeing.
But it's not one of those maternal effect genes that you knocked
These
out.
are better than that.
But didn't you knock out?
There is one left. There is one left in the system.
There's one left.
There is one left.
Okay.
That is something is very hard to remove because this is already 4 mutant and 2 of your live readout in the system. So it becomes, there's one left. And the way it works, Bitcoin works as a translational repressor for that. So Bitcoin could be also present in the cytoplasm, very low concentrations still. And now that binds on the specific sequences of, I think, 3' UTR of this gene called cardin.
It's called, yeah.
Yes. So it suppresses that. So in these embryos, still we have, depending upon whether you are low level of Bitcoin, then still you have a problem. So we thought this is empty embryo means there is nothing left. But that's not the detail we after found out after 2 years of experiment. There is something else left that is fertile and that can work as well. Yeah.
So if I understand correctly, the PCT axis consists of that there's a reaction thing.
Yes.
You can rely on the PCT axis is cell part, right?
Yes, it will be. Yes.
So, so if you turn on the blue light, moving the PCT axis is pretty fast.
Yeah.
Right? If you
Input is also fast.
But if you shift from blue light to dark, right?
Yeah.
Coming back of the PCT axis,
It's
depends on its own development.
yeah.
And from the video seems to be fast. But for another purpose, right?
Yeah.
So if you don't test more proteins together, if you want to test all 4